Okay, for this lecture, we are going to talk about genomic imprinting, aneuploidy due to non-disjunction, and uniparental disomy, and a couple other things. Okay, so let's start with genomic imprinting. Okay, so for genomic imprinting, you will remember that you receive one chromosome from mom and one chromosome from dad. And we're going to look at two different chromosomes. So we're going to call them chromosome 1 and chromosome 2. And if a gene has a maternal imprint, that means it's off on the maternal chromosome, so off from mom. All right, so we'll say this one has a maternal imprint, and chromosome 2, we'll say, is paternally imprinted. And so the way I represent that is off in mom, on in dad, and for the paternal imprint, off in dad, on in mom. All right, so this is happening in you. Okay, so all of your chromosomes will have the same maternal imprints. All of your cells will have the same maternal imprints, for this case on chromosome 1, and paternal on chromosome 2. Okay. Imprinting is on autosomes. So we're not talking about sex chromosomes at all. This is all on the autosomes. Okay, so let's say that you are a female. All right? So in your eggs, which is your reproductive cells, you are going to reset imprints for the maternal pattern. Okay. That means in your germline cells that are going to produce your gametes, okay, your chromosomes will be reset so that they have the maternal imprint. So this is chromosome set 1 and 2. So on chromosome 1, maternal is off. That means on both of yours now, chromosome 1, maternal will be off, because that's how it is for females. And for paternal, on chromosome 2, it's expressed on the moms, and now you are the mom. So both of your chromosome 2s will be on, right? So that your eggs, because you're the mom, we're going to draw both your chromosomes as red to help keep track. Your eggs will have chromosome 1 off and chromosome 2 on. Right? So let's do this now. Oops. Yeah. This might not work. Let's do this now for you being male. Okay, so we have mom up there. Now you are male, which means you're going to produce sperm and reset the imprints for the paternal pattern because you are the father. So again, your germline cells, you're going to erase those imprints and reset them for dad. So that means they're both expressed from chromosome 1 and they're off chromosome 2. So now your sperm will have chromosome 1 on, chromosome 2 off. So that if we combined these with the maternal, right? I don't know if I can do this. Let's see. No, I don't think so. You will have red, which is 1 off, 2 on, 1 off. Now let's draw this a little bigger. Hold on. Red, 1 off, 2 on. And your little zygote, let's make this green for the new baby. The new zygote is going to have from dad 1 on, 2 off, 1 on, 2 off. Right? So they have the same complement that's normal. So maternal imprinting, genomic imprinting is reset in a person's gametes based on their sex. So if they're producing egg whoa, I don't know what I just did. Egg or sperm. Okay. What you inherit as a zygote remains in all your somatic cells. So all the other cells of your body. Okay. So that's the basis of genomic imprinting and how it is reset. So let's look at a situation where we have a mutation in an imprinted gene. Okay. So here's your cells. Here's your dad, chromosomes from your dad, chromosomes from your mom. This is set 1, this is set 2, and we know that it's inherited like this. So let's pretend that there was a mutation here in the dads. Okay, so this green is going to represent a mutation. So this is your phenotype, right? You inherited from your dad a mutation in that imprinted gene. Let's talk about the effects on the offspring. So in your gametes. So if you are female, right, you're going to reset the gametes. Let's add this mutation. This is group 1 and group 2. So you're going to reset the gametes for females, which means for chromosome 1, it's off. And for chromosome 2, they're going to both be on, right? Because you're resetting. So that means for your children, the mutation you inherited from your dad will not affect your children. The mutation you inherited from your dad will not affect the next generation because it's off. Right? So you expressed the phenotype because the mutation was on in your DNA, but in your children's, that mutation is turned off anyways due to genomic imprinting. Now, on the other hand, gametes for a male, okay, so this is in your germline cells, the gamete producing. Let's draw his. So we have mom chromosome, dad chromosome, chromosome 1, chromosome 2, mutation. So for chromosome 2, it's a paternal imprint, so both of these genes are off. I mean, these genes on both chromosome 2s are off. For chromosome 1, it's a maternal imprint, so dad is going to turn on that gene, which means half of his children will be affected. Right? Because half of them will inherit this chromosome 1 with the mutation. So draw that out, say for if you had a mutation in this maternal gene here, and if you were a female, if you were a male. Okay. So again, imprinting is reset in, and I know these aren't, I shouldn't say gametes. These are the germline cells. Oh, why can't I erase that? Because your gametes, the sperm, is only going to have half, right? The information, so it may get a normal chromosome 1, or it may get a mutated chromosome 1. Okay. The reason imprinting is so interesting, and you look at this inheritance pattern, is because remember that imprinting is considered monoallelic expression. So you're only expressing from one chromosome. So let's see. The gene on one chromosome is expressed, and the other one is silent. So you don't have, technically, you have two copies, but only one can be expressed. So we call it monoallelic expression. So that changes when you're looking at inheritance compared to typical Mendelian inheritance. So practice drawing out imprinting. Okay, let's switch now to aneuploidy due to non-disjunction. So due to incorrect segregation of the chromosomes. So let's say you're heterozygous for the A allele, right? So normally, I should make this a little bit smaller. Draw. Normally you would undergo S phase, which is DNA replication. You would get your chromosomes replicated. Oops, that's not correct. Okay. And Meiosis One, you split the homologous pairs, and Meiosis Two, let me just write this, split homologous pairs. Meiosis Two, you split the sisters. So if it helps you, don't hesitate to draw those spindle fibers so you know where they're going. Now you're splitting the sisters. Let's see if I can fit it on here. Right, so they each are gametes, so they have half the number of chromosomes as what you started with, and each cell has one of each, or has one A chromosome. So this is normal meiosis. And why it keeps doing that. All right, let's go to non-disjunction we'll first do it for Meiosis One. So let's say we've replicated the chromosomes, okay, and we're going to have non-disjunction in Meiosis One, which means that incorrectly both homologous pairs go to one cell. So you have one cell with no A chromosome, and you have one cell with both connected homologous chromosomes, or I'm sorry, both homologous chromosomes, and then you split it regularly in Meiosis 2, right, which gives you, two cells with n+1. And this cell that didn't have any is a n-1. So it doesn't mean they don't have any chromosomes, just not the one we're following, right? So if this was humans, they would still have 22 of the other ones. And this would be 22 also, 23, 24. So normal would be 23. That's why you have n+1, n-1. Okay, so non-disjunction Meiosis 1, the homologous pairs stay together. And what I want you to see is that your genotypes, right, are back to this heterozygous, right? It should be big A or little a, but now it has one of each. And this is different than if you had non-disjunction in meiosis 2. So, if meiosis 1 happens properly, right? Let's just move it all the way over here. If meiosis 1 happens properly, you've split your homologous pairs. Right? Now, if you have non-disjunction in meiosis 2, we're going to have two options. So it could happen like this, where for some reason both big A's go together. So this cell, it's happening normally. This cell, you get both big A chromosomes. So this is AA and plus 1 and minus 1, and this would be normal. Okay, or you could have non-disjunction meiosis 2 in the little a cells, right? So that means these would separate normally, and this one would produce a gamete with n minus 1, n plus 1, and it has an AA, n, and n. So non-disjunction meiosis 2, what I want you to see is if your chromosomes are different, in this case like in heterozygous, if it's non-disjunction in meiosis 1, you end up with the duplicate of the same chromosome, versus here you end up with one of each different chromosome in non-disjunction meiosis 1. Okay? So this is a little shortcut or a hint way to double check yourself. If it's heterozygous up here, non-disjunction in meiosis 1 will also produce this heterozygote n plus 1, but non-disjunction in meiosis 2 is going to produce a homozygous n plus 1. Let's try that for sex chromosomes. So let's do that for a male who's X and Y. So we're going to duplicate the chromosomes. Okay. If we had non-disjunction in meiosis 1, one cell would get all of the sex chromosomes. And what I'm doing is I'm setting them up to divide, so metaphase meiosis 2. And then meiosis 2 divides normally, so you've got an X, Y, and X, Y gamete, and then you have two gametes with no sex chromosomes. Okay. Two different sex chromosomes, and we could put the same n, let's do it in a different color. This is n minus 1, these are n plus 1. Okay. Let's say non-disjunction happened in meiosis 2. So we duplicate the chromosomes. They split properly the first time. And then you have non-disjunction meiosis 2. Let's do it first for the X chromosome. So we're going to assume the Y's split properly. So you have your Y chromosome, your Y chromosome. So these are normal Y, normal Y. This one is N-1, doesn't have a sex chromosome, and this chromosome has 2 X's, right? Or you could have Nondisjunction Meiosis 2 for the Y. So that means the X, this cell with the X chromosomes splits normally, so you have a normal N haploid X sperm. I think these little spermies. But you could end up with a sperm that has, I'm running out of room. Hold on. Why this thing doesn't like me. Bigger. N-1, and this one has 2 Y chromosomes. So again, they're not really heterozygous, but you have 2 different chromosomes. You have XY, and the only way you're going to get one of each of those in a gamete is through Nondisjunction Meiosis 1. Okay. If you have Nondisjunction Meiosis 2, you can get 2 X's or 2 Y's. I hope you're kind of seeing that pattern. Okay. Let's do it one more time with a female. So she's XX, but let's say there's a mutation or something in this X. So we're going to color it green. And what I want you to know is we will not take into account X inactivation when doing non-disjunction. That gets too confusing, and we'll talk about in a minute. The X chromosomes are both reactivated anyways, so do not take X inactivation into account for non-disjunction. Okay, so we have her 2 X chromosomes that have been replicated. Non-disjunction Meiosis 1 is going to keep them together. Sorry, I'm getting sloppy here. And you will have 2 eggs without sex chromosomes, and you could have 2 eggs with 2 X chromosomes, 1 of each. Non-disjunction Meiosis 2, right? We're going to split those X chromosome homologous pairs properly the first time, okay. And then you could get either again, normal, or whatever that, if there's a mutation, N+1 and -1, or you could get these doing non-disjunction. Oh, I shouldn't change all those colors. 4 XX XX. So say there was a mutation on the green X chromosome. You can see that if you have non-disjunction in meiosis 1, you're going to get one of each. Non-disjunction in meiosis 2 is the only way you could get both mutated X chromosomes in the same egg. Okay, so that is non-disjunction. So I personally have to draw it out every time. I would suggest that you, when you're going through the extra credit, you really draw these out so you can see how you can get the right answers and why some of the other answers are incorrect. Now, I have another blank. I want to go, I'm going to jump, move these guys up. Okay, so let's talk about when X chromosomes are reset. Okay. So X inactivation, random X inactivation happens in the embryonic tissue. So let's back up a minute. What we believe happens is, if you are a female, okay, and remember X inactivation only affects females or people with more than one X chromosome. Okay, so let's keep it simple and say you are a female, you are XX. And yes, one of your chromosomes were inactivated. But in your egg, both X chromosomes are active in the egg. Okay. For males, which are XY, so they have no X inactivation. What we believe is that the X chromosome is actually inactivated in the sperm. Oops, that's not going to help me. X chromosome inactivated in the sperm. Okay. So here we have fertilization happening. The zygote has one active, and this is going to be a female. If we keep it simple, it could be an XXY male. The male's X chromosome is inactive. That's the blue and orange spots. And it stays inactive in this extra embryonic tissue. So this is the part that's making the placenta here. Okay. But in the cells that are going to become the embryo, okay, you reactivate the X chromosome and then you randomly inactivate X parental or X maternal. Okay. So let's talk about this again. A zygote starts dividing and forms some tissue that's going to become part of the placenta, which is, we call it extra embryonic. It's not going to become the little baby. Okay. In those, X chromosome from the dad is inactivated. Okay, so it still has a it still has an X chromosome active from the mom. In the tissue that's going to become the embryo, the paternal X chromosome is reactivated, and then you get random X chromosome inactivation, so that females are a mosaic of, I'm not drawing this very well, but the paternal and maternal X chromosomes. I have one more slide that shows the same thing, just slightly different. Okay. Again, zygote, you have paternal X chromosome inactivation. That's going to stay in these orange cells. Okay. Imprinted, the paternal X, in the cells that are going to make, so ES stands for embryonic stem cells, the cells that are going to make the embryo. You have reactivated the paternal X chromosome. You now have 2 functional X chromosomes. You have random X inactivation. This is trying to show you these are going to be the embryonic cells. Make this little red embryo. And then this is showing you in the egg making cells, right? We're going to have both X chromosomes inactive again because let me start again. In the egg making cells, we are going to have both X chromosomes active again because those are going to be activated to pass on into your eggs to pass on to the next offspring. Okay, so random X inactivation happens in the embryonic tissue. In the egg-making cells, both X chromosomes are activated, which means you can pass on the X chromosome from your mom or from your dad. All right, let's see what else I got for you. Okay, uniparental disomy. Means that both chromosomes a person has, or in that cell, of a pair, right? This could happen with the pairs 1 through 22 is from the same parent. Okay, so we have meiosis happening, and this is showing non-disjunction in meiosis 2. Right? And so we've drawn this out before where you could have 2 chromosomes from that one parent in a gamete. Fertilization means, say these are eggs, you've added the sperm. So that's where this 3rd chromosome comes from. And many times you can have what's called trisomy rescue, where you have another weird kind of a non-disjunction event and you lose 1 chromosome. Okay. So now you're back to 2 chromosomes, but in this case they're both from one parent. So this cell has uniparental disomy. One of the things I want you to see is that you still have 2 copies of each gene. Right? And so, I can't. Sometimes I can zoom. This is why one of the questions talked about where you would have gene dosage issues. You're not going to have gene dosage issues here because you still have 2 copies of the gene. Okay. So you still can have biallelic expression. Where this becomes an issue is, say, with imprinting. Say this gene is off, we're going to pretend the purple is mom and on in the dad. And so when you lose that father's chromosome, now you have no copies of that being expressed. So imprinting can, what do I want to say? Can affect gene dosage in uniparental disomy. But for regular genes, you're going to still have 2 copies of that gene. We don't know if those are 2 dominant, 2 recessive, 2 mutant, 2 whatever types of genes, but you would still have 2 expressed. So let's look at another slide. Okay, so here we go. We start off and we produce gametes, and one has had a non-disjunction event. When fertilization happens, so you're not going to get trisomy until you get fertilization. Now we have trisomy. We have 1 from dad, 2 from mom. Rescue, which we just talked about, means the loss of 1 chromosome. And it's rescue because you're now back to the normal diploid state. Could happen a couple different ways, so let's put a blue mark here. So you could lose the red with blue chromosome, so this goes off to Never Never Land and is gone, right? So now you're normal. Or you might have the blue and the red with the blue mark. Normal, you got 2 chromosomes. But if you lose the male, the blue chromosome, now you have uniparental disomy because you have 2 chromosomes that came from the same parent. The last slide for this uniparental disomy shows a couple different ways this can happen. Okay, so we talked about trisomy correction or rescue, where depending on which chromosome you lose, you could have uniparental disomy, or you could be normal, having 1 chromosome from each parent. Okay. Monosomy duplication means that 1 gamete has no chromosome for whatever that pair is, right? So when you fertilize, your zygote only has 1 copy of that chromosome. Monosomy rescue means that after duplication, after S phase, you have 2 copies, right? Like this. And for some reason, you have another non-disjunction, and now both copies are staying in the cell. This is called uniparental disomy because you have 2 copies of the same chromosome from the same parent, right? And you could switch these, right? This could be an egg and a sperm with nothing, and you'd have maternal uniparental disomy. Gamete complementation is talking about having 2 already, right? So non-disjunction happening again twice, nothing from this parent, right? But whoa, oh, this started doing this to me the other night. Hold on a second. Knew it was too good to, be working for a while so long. Okay. Uniparental disomy in this case is because you already inherited 2 from another parent. Nothing from this parent you don't technically have to rescue it or have a duplication. And again, this could be 2 in the sperm, nothing in the egg, and that would be paternal uniparental disomy. So lots of different ways it can arise. All right, the final is just a reminder of cell cycle. Okay, I kind of assumed people remembered this. S phase is where we're talking about DNA replication. So G1 is where the cell is growing, preparing to replicate. Here is where your licensing factors for DNA replication are put on. Remember, licensing factors for DNA replication are to allow that DNA replication only happens one time in S phase. Okay. After your chromosomes are duplicated, your cell is going through a G2 phase where it's double checking, making sure you are ready to split, and then this could be meiosis, or you could go into meiosis and eventually produce 4 gametes. So, you hear me talking about S phase, G1 is before S phase, G2 is after S phase, S phase is where the chromosomes are duplicated. All right, I hope that helps you with some of these tough concepts. And that's all I got for you.